In this lecture, I'm going to cover the concepts regarding the Sordaria lab, which has to do with meiosis and sexual reproduction of a certain type of fungus. So in this lab, we're going to allow sexual reproduction to occur between two different types of fungi. We're going to see crossing over take place, the results of crossing over. And then the results of crossing over are going to help us actually figure out where a specific gene is located on the chromosome of this type of fungi. So to understand the concepts of this lab, there's really two big ideas we first have to discuss. First, we have to discuss sexual reproduction in this type of fungus, which is called Sordaria. It's a type of ascocarp. You don't have to have that memorized, but there's many different types of fungus. And we're going to see that their way of sexually reproducing is a lot different from the way that we sexually reproduce. They actually have to utilize both meiosis and mitosis to make reproductive structures. And we're going to see the main reproductive structure we're going to talk about is called the asci. I'm going to draw one for you here in a little bit. And inside the asci are going to be the next generation. We're going to call them spores. So wherever these spores ultimately land, then a new fungus will grow. So we're going to talk about sexual reproduction first, and then after we get through the specifics of that, then we're going to talk about linked genes. And again, you have to understand sexual reproduction and linked genes to understand these ideas that we're trying to teach you in this lab. So here's the setup of the lab. We're going to take an agar plate, and we're going to put two different types of Sordaria fungus on them. One type has the ability to produce tan spores, and the other type has the ability to produce dark spores. And how we do this is I've sent samples of these two different strains on agar, and I just cut out a piece of the agar and I just place it here, place another one here, and this is from the strain that has those dark spores, and that's why that square is filled in dark. And then I cut out a sample from the fungi that can produce tan spores, and I put a sample here and I put a sample here, and the fungus is going to grow. Now before I show you that, I want to indicate what this plus means, because we're going to see this further on in the unit and in many genetic problems. When you see a phenotype like dark spores and it has a plus associated with it, then that means that that phenotype is the most common one in the population, and it's called the wild type. Now, it doesn't mean that dark is dominant and tan is recessive if it's the wild type and if it's the most common one. It just means that it's the most common. As a matter of fact, sometimes the most common phenotype or the wild type could be the recessive allele. So it just means it's the most common phenotype, and we put a plus to associate it with that phenotype. Whenever we have a rare phenotype, and it's usually due to a mutation, then we'll put a symbol for it, and lots of times we put a negative with it. So this is the symbol that means the phenotype tan, and it's due to a mutation, and it's pretty rare in the population. So here's what's going to happen in this agar plate, is the fungi body is going to start to grow by mitosis. And what it looks like is you see these little fingers that form here in the body of the fungus. And each little finger is called a hyphae. And we're going to see that the hyphae from one strain will meet with the hyphae of the other, like you see right here. And if the two hyphae fuse from two different varieties from two different parents, then this is sexual reproduction. We're going to see here in these areas, we're going to see spores form, and it's due to sexual reproduction. Now we're also going to get asexual reproduction in these areas right here. So I'm just going to put asexual, 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 asexual. And that happens if two hyphae from the same parent meets. So I'm just going to show, I shouldn't erase the whole thing, but imagine if these two would meet and fuse, then we're going to get reproduction, but it's due to just one parent. So we're going to see both sexual and asexual reproduction. So in the agar plate, it's really hard to kind of see what's going on there as the hyphae are fusing and how this is related to sexual reproduction. So we're going to draw it out here in the space below. So let's show hyphae from two different strains meeting. So again, they look like little fingers here. And in these cells that are about ready to fuse of the hyphae, there's a nucleus Let's draw the nucleus in here. And we know that our body cells are diploid. They're all 2N. And then we make these haploid gametes. And we kind of figured that's the way it is for all organisms. But for a lot of organisms, that is definitely not the case. If you look at the body cells of fungi, they're not 2N. They're not diploid. They're haploid. They're N. So let's indicate that. And remember, we have 2 different strains here. And to kind of symbolize that there's going to be a chromosome that codes for dark spores in this nuclei, I'm going to color it dark. This one has the chromosome that's going to code for the tan nuclei, so I'm going to leave it light there. So let's show that there's fusing, and let's kind of indicate that first the cell membranes are going to fuse, and then we're going to see the nuclei is going to fuse. So we're going to kind of get one big long structure here after the cell membrane fuses, and we're going to end up with just one nuclei because these are going to end up fusing with one another. And to indicate that we're going to have a chromosome for dark and a chromosome for light, I'm just going to color in the nuclei like that. Now, anytime we add 2 haploid nuclei, N plus N, now this nuclei is diploid. It's 2N. And from here on, this is going to look really similar to a diploid cell of ours, a germ cell, dividing by meiosis to give us 4 daughter cells. So again, at the end of meiosis here, you're going to see 4 nuclei, or like 4 cells. Now, between here and here, our germ cells, they're going to go through S phase and replicate DNA. Same with this nuclei. We're going to see replication of DNA. So we might indicate from here to here, we're going to get replication, DNA replication. Okay, so technically, then we're going to end up with a nuclei that's 4N. You want to do some math there. You have a 2N nuclei and you double its DNA, then it's 4N. Ours is 4N. Our cell is 4N in G2 before it goes either into mitosis or meiosis. So from here to here, let's start meiosis 1. Remember, in meiosis 1, we go from 1 cells to 2 cells. It's called the reduction division. So meiosis 1 is going to take place. So meiosis 1. And now, in this structure, we're going to end up with 2 nuclei, because this one splits from 1 to 2 in meiosis. And now, do the math. Those would be 2N. Let's go on to meiosis 2. So let's indicate meiosis 2. And so then this one's going to split into 2, and this one's going to split into 2, because we're going to end up with 4 cells, or 4 nuclei. So this one splits into 2, so let's show 2 there. And this one will split into 2, so let's show 2 here. And let's go back and color code. So this one gets a chromosome for tan spores, this one gets a chromosome for dark spores. When this one splits, tan, tan, dark, dark. So this looks really similar to our meiosis. We started out with a diploid cell, replicated the DNA. In meiosis, you go from 1 cell or 1 nuclei to 4. We know that at the end of meiosis, all of the nuclei, or all the cells, or all the gametes are haploid. So let's indicate they're all haploid. But their sexual reproduction is not done yet because they have another division after meiosis. They have a division of mitosis. So remember, in mitosis, we go from 1 nuclei to 2. So what's going to happen is this nuclei is going to split and split. So we're going to get 2 nuclei from it. Let's show this one. Let's show some arrows here. This is going to divide by mitosis. And this is going to divide by mitosis. Okay. And I also should probably indicate that before it splits by mitosis, it does copy its DNA. Otherwise, we wouldn't end up with the haploid N cells that we get here at the very end. Remember, in mitosis, you get 2 daughter cells. They look identical to the parent cell. The original nucleus, the original cell was N. Then both of the daughter cells are going to be N as well. So from here to here was mitosis. So let's go ahead and color code them in. And we're going to indicate that all are N. So all these are haploid spores. And if one of them lands on the ground, then it's going to start to divide by mitosis, and it's going to grow into the body of one of these strains of fungi. So that's how they sexually reproduce. So ultimately, in the end, that last division, mitosis, allowed 8 cells or 8 spores to form, and they're going to be haploid. Now, let's talk about very last diagram right here, and really what this looks like. This develops into this long sac here. Let's put the spores in there. Again, they're all haploid. And the whole thing is called the asci. But each individual little circle here is the spore. So when I'm talking about the asci, that's what I'm talking about. It's kind of these long little packages there. It kind of reminds me of sixlets. So one sixlet package is like an asci. Now, I'm going to show you this again, but now we've zoomed in and you can actually see the chromosomes that are coming from the 2 different parents, the 2 different types of fungi. And you have to be able to look at different diagrams and be able to tell what's going on. To be able to tell me like which parts of the diagram would be showing you meiosis, which part would be showing mitosis, and so on. So I'm going to add again kind of that initial step. I'm going to show one hyphae and another hyphae growing together here. And they each have a nucleus and they each have a chromosome that contains a gene for the spore color. And of course, they're color coded for you because they're trying to show you that this chromosome, like right here, we can say has an allele for dark spores. This one here is going to have an allele for tan. So let's label some of these events that we just diagrammed on the previous page. So from here to here, we can tell that replication was occurring. And this diagram is just showing you after these 2 have fused. So let's show that they fused, and this is what that nuclei looks like. From here to here, from 1 cell to 2 cells, that would be meiosis 1. From here to here, here to here, this is meiosis 2. So we've completed meiosis. So we went from 1 nuclei to 4. And remember, at the end of meiosis, the nuclei should be haploid. So let's say these are all in. Then there's that final division, mitosis happens. And again, we end up with parent cells that look identical to the daughter cells. So see, these daughter cells look just like the parent cell. And you go from 1 nuclei or 1 cell to 2. So this step is mitosis here. And again, it's showing you that these are going to be darker spores. So I'm going to fill them in here because they all contain a chromosome that contains an allele that codes for those dark spores. Now, before I move on to the next diagram, you can see right here, whenever the homologs pair up, just like they do during meiosis when our cells are dividing, there was no crossing over. Because they didn't swap pieces. If they did, then we would see this chromosome down here would have like a lighter part to it, where it accepted a piece from this chromosome. So we can tell no crossing over occurred. Now, when you're doing this lab, you're going to have this memorized, basically. If crossover does not happen during sexual reproduction of these 2 types of fungi, then we're going to see these asci form, and they're going to have a 4 and 4 pattern. We're going to see 4 dark spores together in an asci, and there'll be 4 light. And you can see why that happens. There was no crossover. So we get the light ones all together, and we get the dark ones all together. Okay? So we're going to look for asci that look like this, and when we see them, we're going to mark them as no crossover happened. This is just another diagram that's basically showing you if no crossover occurs, then these are the patterns that we're going to get here. Okay? Now let's take a look at another diagram, and it's going to look different because I need you to get familiar with different diagrams showing these fungi dividing and sexually reproducing, because I have no idea what the diagram might look like if they show this to you on an AP exam. So let's take a look at kind of a different one here. So the first one that I have here is showing you if no crossover occurs, we're going to get that pattern that we just mentioned, 4 and 4. So look how they show the chromosomes here. They're indicating that this one is coding for those tan spores. And this chromosome that's already been replicated because they look like X's, it's coding for those dark spores. And as you follow this along and you see meiosis and then mitosis and so on, you can see that there's no crossover. No crossovers occur. So let's take a look at these other diagrams. So if we get any other arrangement of light and dark, like we see here, that's not 4 and 4, then you have to know that crossing over did occur. So let's take a look at how they're diagramming this. So we definitely can see crossover is happening right here. And so this chromosome has a code for dark, and this chromosome has a code for light. This chromosome's getting that allele for dark, and this chromosome has the allele for light. And when they're sorted out through meiosis and mitosis, then we're going to have an allele here going into this spore, and it's coding for dark, dark, light, light, dark, dark, light, light. And you can take a look at the other diagrams because it's just showing you different ways that these homologs can cross over. And due to these different ways they can cross over, we get these different patterns. Okay? So basically, any pattern other than a 4 or 4 is due to crossing over that has taken place. So whenever we're analyzing our fungi and the spores that we're going to grow in that petri dish, we're going to be looking in the microscope and we're going to be seeing the ASCII. We're going to be seeing things like this. And ultimately, all you have to do is you have to figure out the percent of the ASCII that are showing crossover. So it helps if you start at 12 o'clock and you make some marks as you go around the clock. So I'm going to put an N for no crossover. So 4 and 4, no crossover, no crossover. And you can hit pause and try it yourself here in just a second and to see if we come up with the same numbers. From this, I've determined that there were 13 ASCII that showed crossover. I have 10 N's there because they were not showing crossover, so 10 did not. Again, I'm trying to figure out the percent of the ASCII showing crossover, so I just need to take 13 divided by my total there, which is 23, and then that's going to give me my percent, which is about 56.5% after I times by 100. Now, I'm going to pause there because this is an important number. Here's our answer. We figured out the crossover percent and the offspring. There's going to be more that we're going to do with this number, but we have to go and learn about kind of the second part of this lab, or the second concept related to this lab, and that is linked genes. When I'm talking about linked genes, I'm talking about genes that are found on the same chromosome. And since they're on the same chromosome, there's going to be a distance between them, and that distance is measured in something that we call map units. So we can measure the distance between two genes in map units. We can also measure the distance between a gene and its centromere. So let's just do some simple practice. I have a diagram over here, and then we have these two questions. Question A says, how many map units is the UNC75 gene from the centromere? So note that the centromere is at 0 here. Here's the UNC75 gene. It's right before this 10 marker. So I'm going to go with 9 map units from the centromere. And sometimes we just put 10 MU, map units. How many map units apart is the MAB20 gene and the UNC75 gene? So again, here's our UNC75, and let's look for our MAB20, or MOB20, I believe. So MOB20 is right here. Okay. So it looks like this one is about 11 from the centromere. And since we just said this one is 9 from the centromere, then that means that they're 20 MUs apart. Now we know that if we have genes that are linked, which means that they're on the same chromosome, like we see here, gene A, B, and C are all on the same chromosome, then we know that we can see crossing over occur between the homologous chromosomes. And what that leads to is kind of new combinations, new chromosomes. And we have a term for them. They're called recombinant chromosomes. So here we see crossover is occurring, and these two are considered the recombinants, and these two are non-recombinants. Now, the individuals that inherit these chromosomes, sometimes we also call them the recombinants or the non-recombinants. So if an organism inherits this chromosome right here, and it shows these phenotypes, A, B, and C, then we say that organism is a non-recombinant. Those that inherit these and then kind of show some different traits there, those are the recombinants. Now here's a couple rules to remember about linked genes. If genes are farther apart from each other, or if the gene is a long distance from the chromosome, then crossing over is more likely to happen. Now let's think about that. Here I have the location, the locus of gene A and gene B, and then I have gene A again over here, and I have gene C. So let's determine which two alleles would show crossover more often. Well, if you think about, to cross over gene A and gene B, we're going to have to break the chromosome here. And that's kind of the only area where we can break the chromosome to get crossover. So we're going to see crossover less often between gene A and gene B. Whereas if we look at gene A and gene C, we can break the chromosome here, and we'll get crossover, or if we find a chiasma forming here, we'll get crossover, or here, or here, or here, or here, and so on. I think you get the point. So we're going to see crossover more often because those genes are further apart from one another. So let's say that if we see crossover happen in offspring, and we see it happen 5% of the time, then that means that those two genes are 5 map units apart. If we see crossover happen 10% of the time, the two genes are 10 map units apart. If we see crossover happen 33% of the time, the two genes are 33 map units apart. I think you can see where we're going with this. So that's something that you need to have memorized. If I say that we see recombination 12% of the time, recombination means crossing over, then that means that the two genes that are showing recombination are 12 map units apart. So let's look at some of these practice questions. It says, which two genes would show recombination more often? A and B or A and C? We already determined that would be A and C because they're further apart from one another. If gene A and B cross over approximately 5% of the time, then we already stated gene A and B are going to be 5 map units apart. Gene A and C show recombination 22% of the time, 22 map units apart. And you just need to remember that recombination equals crossing over, means the same thing. Gene A shows crossover approximately 7% of the time. Gene A is how many map units from the centromere? Notice in these problems, we're not really mentioning a second gene. We're just talking about one gene here, gene A. There's no mention of gene B, gene C, or anything. So in this situation, we're just seeing how far away gene A is from the centromere. So if we see crossover for gene A approximately 7% of the time, then we say that gene A is 7 map units from the centromere of the chromosome. Gene C shows recombination 29% of the time. Gene C is 29 map units from the centromere. So again, very simple problems, but very confusing if you've never seen them before. All right, so here's a few more practices for you. So take a second, pause, and you can come back and you can check yourself. So for E, we really don't need to reference the diagram to answer the questions because it says that if we have two genes, they're 5 map units apart, we're going to see crossover happen 5% of the time. If these two genes are 5 map units across, then that means, it's just another way to say it, we're going to see recombinant offspring crossover 5% of the time. What percent of the offspring will be recombinants due to crossing over between this GLP gene and the FOG gene? So we have to find those here because we have to figure out how many map units apart they are. So from here to here, there's 5 map units. And so from here to here, I would go with that being about 21 since it's not right at that 20 mark there. So we're going to see 26% of the offspring are going to show the recombinant phenotypes because those two genes are 26 map units apart. H says the EGL gene crosses over 14% during meiosis. This is just another way to say it. I'm just trying to expose you the different ways that they can say this or ask these questions. So the distance for this gene from the centromere is going to be 14 in use, map units. So we just determined that crossover frequency equals map units, the distance between two genes. So because of that, researchers can use this to estimate the distance between two genes. We can figure out how far a gene is from the centromere, and then we can also figure out where different genes are located along the chromosome. And that's what these next two problems are about, is you're going to determine the location of different genes due to their crossover percents that we see in offspring. So here we have three different genes, and we're trying to figure out their positions on a chromosome here. It says that they're linked, and it says here's the crossover frequencies, and they're showing you the crossover between the different genes that we're analyzing here. When I tackle these problems, I like to draw out the genes and indicate how far apart they are. So I'm going to draw a big chromosome here, and it says that B and D genes show crossover 5% of the time. So I'm going to put them here, and I'm going to put them pretty close together. I'm going to indicate from here to here is 5. And let's say this is B and this is D. My next clue, it says B and A is 30. So I could maybe go 30 this way, and that could be A. Or I could go 30 this way, and that could be A. So I'm trying to figure out where A is at. I'm going to put a question mark there because I don't know exactly where A is at. When I look at the next clue, then it tells me which of those two locations I would find the A. The next clue says DNA is 25% apart. So I know that this has to be my location of A. They just said that B and A is 30 apart. So since this whole line from here to here is 30, and this much of it was 5, then that means that this much from here to here would be 25. Just like they say, DNA is 25%. If I had put A over here, and remember they said that B and A was 30, so they're saying from here to here was 30, then that would actually put DNA 35 map units apart, and it's not, it's 25. So this is just a little bit of trial and error. You kind of draw different things, different locations where you could find different genes, and then you see if basically the math fits where you think these different genes are going to be located on the chromosome. Why don't you hit pause and try J on your own, and then I'll show you the correct answer. So now we have 4 genes, and we're trying to find their relative locations, so they're probably not going to be in A, B, C, D order. It says that A crosses over with C 20% of the time. So let's go ahead and draw my chromosome here. And I'm going to kind of put them in the middle because I don't know exactly how far we're going to have to move down this chromosome. So A and C 20%. So let me put A here, and let's put C here, and let's indicate 20 map units. The next clue says that B crosses over with C 15% of the time, so I'm going to put a mark here. And I'm going to put B might be here, or we might go this way. And we might put B here. And again, from C to B, we've got 15. And of course, from here to here would be 15. So I'm going to mark that line 15. Next one says A crosses over with D 10% of the time. So that means that we have to go 10 away from A. So I'm going to split this 20 here. I'm going to put a D here, question mark, not sure. Or we might be going this way, 10, and put a D here, question mark. So I might indicate there is 10 there between D and A. And then finally it says, and D crosses over with B 5% of the time. Well, let's just take a look at the locations that I have here with question marks for the D and the B. And we just indicated that they're pretty close together. So D can't be here and B can't be here because then they would be 25 map units apart. So they're only 5. This is the distance from here to here. That would be 5. So I can go ahead and get rid of, erase, to the best of my ability here, the locations of these question marks, and I figured it out. So on this chromosome, I would say that the order is A, then goes B, then goes D, then goes C. And remember, it just depends on which end of the chromosome you start off from when you're reading it. You could also say the answer is C, D, B, A. Just go backwards. We went through sexual reproduction of Sordaria. We talked about what does it mean if genes are linked, and what does crossing over tell us about the distance between genes, or the distance between genes and the centromere. So now let's go back to the results of our Sordaria lab. So I've already shown you this picture before, and we already counted up, and we determined that crossing over happened 56.5% of the time. Now, some of you maybe put 2 and 2 together, and you determined, hey, we have a percent of crossover that took place in the Sordaria. So the gene that codes for spore color is 56.5 map units from the centromere. And that would be correct, except we're dealing with fungus, and they reproduce a little bit differently. And they have that extra division there by mitosis. So we actually have to correct this percent. We have to divide this percent by 2 to get the corrected crossover percent, because only half of the spores in each ascus are the result of crossing over.